Okay, thank you, Brian. And just bear with me on my voice. I am just on the very tail end of a cold, but we all know how these things linger, so I will try to speak up as well as possible. So I'm happy to be here today to talk about some of the studies we've done, combining two kind of different applications that we've developed separately using siRNA and HEPATOPAC and developing a lipid-loaded model in HEPATOPAC that we found to be very complementary to one another. So let's just start off with some background on HEPATOPAC. HEPATOPAC is BioIVT's long-term hepatic co-culture model. So it's very well established for ADME tox purposes. It was developed to address specifically clearance and MET ID applications for low turnover compounds, and also for getting a more predictive tox profile due to the ability to do repeat dosing. And so some of the function for HEPATOPAC is dependent on the microarchitecture you can see in the middle. It's hepatocyte islands of 500 microns with supporting stromal cells filled in. And this allows for the drug metabolism function to be maintained for up to 28 days with a similar profile to suspension hepatocytes at day 0. But this long-term function, we felt also demonstrated general biological relevance and could be useful for broader applications. Of note, this model also does maintain the distinct morphology of hepatocytes throughout the duration of the culture. So we're looking at the biocanuliculi formation out at 2 weeks. And this is true for all species, so HEPATOPAC is available in both human and preclinical species. So as I mentioned, though this was developed for ADME tox purposes, we know that drug development is a very long process comprised of multiple different stages, and we felt that the HEPATOPAC applications could be expanded to support other aspects of drug discovery. And specifically the fact that we already have this long-term function, the hepatic function, we felt like this was a really useful base for doing more disease modeling and other types of studies that would require long-term hepatic function. So just for instance, as an example, we had a collaborator who we recently tested 2 test compounds in the lipid-loaded model. They had done efficacy and tox studies in HepG2 cells and then jumped over to HEPATOPAC. And so for one of their compounds, they did find that both the tox and efficacy pretty well matched the HepG2 data. However, another one of them, we found significant toxicity at much lower concentrations. And this affected the efficacy for reducing lipid accumulation. So with this model, we were able to both use the metabolic competence, the reason for that being a toxic metabolite, as well as the lipid accumulation. So get both of those data at once. So as I mentioned, this has been extended beyond just ADME tox, and there are a number of applications that HEPATOPAC can be used for, listed here, including infectious disease like HBV, steatosis, as I mentioned, new modality drugs, as well as the standard ADME for small molecules. So I'm not going to talk about all of those today. However, if that is of interest, we do have tons of resources available on the BioIVT website, including training videos, brochures, posters, and our publication compendium, which lists all the, not all of them, but a selection of very relevant peer-reviewed articles that use HEPATOPAC and separates them out by application, so you can easily find whatever you are looking for. So let's jump into fatty liver disease. Just a bit of background on that. Metabolic associated fatty liver disease, if you've been around in the field, you know it had a rebranding a few years ago, MAFLD and MASH instead of NAFLD and NASH. So MAFLD is the earlier stage disease comprised of mainly hepatic steatosis, and that comes about due to dietary factors, such as a diet high in free fatty acids or high sugars, such as fructose. This is often comorbid with diabetes and obesity, and importantly is reversible with lifestyle changes. This can progress as the steatosis builds up, is not resolved. It will induce inflammation, reactive oxygen species, and continued disorder. And then when that occurs, it will progress to MASH, where you now have progressive fibrosis and liver damage. And this is difficult to reverse with current strategies. And I apologize, I do need to update this. When I presented this last, there was one therapeutic that was approved for MAFLD and MASH treatment, Resdifra. There now is approval for semaglutide for MASH as well. But there are still many other targets under investigation for MAFLD and MASH. This is still very much an unmet need in the clinic. So one of the ways that we can start to identify therapeutics for MAFLD and MASH is to use in vitro models. And there are many in vitro models that are available to study MAFLD and MASH. And these often involve adding exogenous free fatty acids, high glucose and fructose, or other molecules, and can combine hepatocytes with other cell types, such as non-parenchymal cells. So the very simplest is a standard 2D culture or co-culture. And I will say that all of these models can have their place and can be useful depending on the application. So the benefits of 2D culture is you can do studies very rapidly. You can load the lipids very quickly. You can add the MPC into the culture. However, as we know with primary hepatocytes, the longevity of that model is kind of limited to shorter term studies. In the middle, we also have 3D microtissues such as spheroids. There certainly is quite a bit of published data on spheroids. They do have long-term viability and function, can incorporate those other cell types, and are very useful for kind of these fatty liver disease applications. And then what I'm going to talk about today is the HEPATOPAC model. So again, also has long-term viability and function. We can incorporate NPC, such as in the hepatimmune model, which incorporates primary human Kupffer cells. One advantage that we find with HEPATOPAC and hepatimmune, the handling of this culture is very simple. So because this is printed onto a standard well plate, you can handle these similarly to a 2D culture. So you don't need any specialized skills to use HEPATOPAC. And in that same vein, because it is patterned flat on the plate, you do not require confocal, microscope for imaging, or any special imaging tools. So we just used a standard high content imager to do a lot of our analyses. This was very useful for getting full well quantification of the lipids. So just to walk through that workflow, you can see the bottom left image is a hepatopac island. Those are the hepatocytes. And you can already see in that bright field image some of the lipids that have accumulated when we treated it with free fatty acids. For these studies, we used a neutral lipid dye, the BODIPI 493503 dye. You can see that in the next panel over in the green stained portions of that image are the lipids. So the HCI analysis software that we used was very nice. It was the Revity Harmony software, and we were able to identify the region of interest where the lipid had accumulated and filter out any background staining. So this is very powerful to get just that lipid portion. And then, as I mentioned, it does take images throughout the entire well and stitches them together. So we were able to get the sum of the lipid accumulation for the entire well. And then it can kind of scan through the entire plate very quickly, so you can get quite a bit of data in a very short amount of time. So just to start, we used some different media formulations to look at lipid accumulation and the time course of that accumulation. So for following from top to bottom, we have control, free fatty acid, high glucose and fructose, or a combination of the high glucose and fructose, and looking from day 2, 4, and 7. So it kind of makes sense that the free fatty acid has very little barrier to accumulate in the cell. It's pretty direct. We saw that the high glucose and fructose did take more time to accumulate in the cell. That is requiring some more de novo lipogenesis, and took about until day 7 to kind of peak. We did find when we switched those cultures back to the lean control media, this was reversible. And it was visible with both the lipid staining as well as triglyceride accumulation. We had also done some assessment of fatty liver-associated genes. So again, cultured with these different media, and used a QIAGEN PCR array profiler for human fatty liver disease, and assessed 84 genes related to human fatty liver disease. And kind of mirroring what we saw with the lipid accumulation, the more complex changes were associated with the high glucose fructose treatment, or the combination of free fatty acid and high glucose fructose. So that's driving kind of a more disease-relevant phenotype. And we've done quite a few applications for both small molecules and new drug modalities. Not necessarily going to talk about all of them here, but looking at kind of repeat dose efficacy, and as I mentioned before, combining that with toxicity, looking at steatosis prevention or reversal, different types of tox, and then what I'm going to talk about today, which is gene knockdown and using siRNA in conjunction with this model. First, just a couple of validation points, and this is using small molecules. We wanted to look at both steatosis prevention and reversal using ACC1 and 2 inhibitors. These are known to reduce lipids, and we did in fact see that that was true, and it was concentration dependent. We also looked at drug-induced steatosis. In these studies, we used valproic acid and used different backgrounds, so either control, free fatty acid treatment, or high glucose fructose. So the cultures were treated with these different medias, and then on top of that, escalating concentrations of valproic acid were used. So you can see with the control, for instance, only the highest concentration induced drug-induced steatosis, and that is well above the Cmax for this compound. Whereas for the free fatty acid and HGF, we saw increased accumulation of lipids, so from the no valproic acid at that very lowest concentration. And that concentration does fall below the clinical Cmax for valproic acid. So this might suggest that the donor background can play a part in the drug-induced steatosis ability of compounds. So with all of that about small molecules, separately, we had developed a gene knockdown platform in HEPATOPAC, and we wanted to combine that with this fatty liver disease model. So using siRNA mainly, and siRNA we had used both as a research tool as well as, it's a drug modality. So why are siRNA being used as drugs? They are highly specific toward their target protein. They have a long-lasting therapeutic effect, and they are less likely to produce drug-drug interactions in the typical way, such as CYP induction or inhibition. That's very target-dependent. And if you look at some of these currently approved siRNA drugs, a lot of them do use the GalNAc delivery method, so that will deliver the compound straight to the liver. As we know, kind of these molecules will accumulate in the liver anyway, but that will direct them just to the hepatocytes. So our objective when developing this siRNA method was just to have effective siRNA knockdown in HEPATOPAC, in the hepatocytes specifically. So I said that this is a co-culture model. It includes those supportive stromal cells, and we did just want to direct this to the hepatocytes. For the transfection portion, we wanted to make sure that we could use commercially available reagents and maximize the durability of knockdown and reduce the toxicity from that transfection reagent as much as possible. And so we know the challenge of getting siRNA into the target cell is that they don't normally cross cell membranes. They're large molecules and also charged molecules, so they need a little bit of help to get into the cell. The first approach that we had used was using a transfection reagent. This was just a lipofectamine-based reagent in our case. So this will trap the siRNA in these liposomes and allow them to be brought into the cell. We know this entry is not cell type specific, and that did kind of factor into the timing of when we do these transfections in HEPATOPAC. So we first tried to do this transfection in mature HEPATOPAC. So HEPATOPAC takes about 7 days to mature and have those drug metabolizing enzymes and functions kind of go back up to baseline. And so doing this transfection at day 7, unfortunately, even with hepatocyte-specific siRNA and genes, those stromal cells were really just kind of sequestering the siRNA. So you can see when we looked at CYP3A4 activity, this was the gene that we were knocking down. We didn't see a ton of knockdown, maybe a 30–40% knockdown, which is certainly not sufficient. So then we took a step back and decided to do the knockdown at day 0. So the way that HEPATOPAC is manufactured, on day 0, we seed the hepatocytes, and then it's not until the next day on day 1 that those supporting stromal cells are added. So we take advantage of this at about 4 hours after seeding, when the hepatocytes have kind of sat down on the plate. We'll do the transfection. So only the hepatocytes are present at that time. And again, we use CYP3A4 as proof of concept, and we did see very good knockdown, about 80%, and long sustenance of that knockdown, at least 18 days. We've got a little bit further, maybe about 21 days. One of the benefits of this as well is it gives a chance for not only the mRNA to be knocked down, but also the protein to be degraded. So we're looking at both mRNA and CYP3A4 activity are both reduced in this model. The second approach is to use modified siRNA, such as GalNAc, to circumvent this issue and deliver the siRNA directly to the hepatocytes. So in these studies, we use GalNAc conjugated siRNA to specifically target the entry into hepatocytes. This is facilitated by the ASGPR1 receptor, which is specifically expressed on hepatocyte membranes. So this could be done at any time during the hepatocyte culture. And we just look at whether there was a correlation between ASGPR expression, the GalNAc siRNA uptake, and gene knockdown. We found a pretty good linear correlation. We do know that the ASGPR expression can vary from donor to donor and at different time points in culture. So this was still dependent on the ASGPR. We did a couple separate studies where we knocked down ASGPR with transfection and then looked at GalNAc uptake. And again, we did find a good correlation, basically just validating that our siRNA was getting into the cell by the expected method. So now combining this with the fatty liver disease model, we wanted to look at different genetic variants that are associated with fatty liver disease risk. And the benefit of this is that For instance, we looked at the PNPLA3 mutation that is associated with increased fatty liver disease risk. We do have genotype donors at BioIVT that we are able to use for these studies. So either wild type or mutant donors were used. And we first just wanted to verify that these donors were behaving as expected. So it's known that these hepatocytes should be more sensitive to lipids than the donors that have the wild type protein, and we found that to be true. If you look on the very, is it the left side for you? The very left side, those are wild type donors. And then right next to it is one of the I-148 genotype donors. That's the increased risk donor. So we treated with escalating concentrations of free fatty acid and essentially saw that those high-risk donors loaded more lipids, and that was true for multiple donors that we combined into one analysis. We also found that when we switched to a lean media in the next panel over, those PNPLA3 I-148M donors had more trouble clearing out that lipid when they were switched to a lean media. So we know that the mutant PNPLA3 is thought to accumulate on lipid droplets to prevent their lipolysis. We wanted to know what happens when you knock down PNPLA3 in genotyped hepatocytes. So for this study, we knocked down, using transfection, PNPLA3 in wild type or mutant donors, and then followed up once the culture was matured with a free fatty acid treatment for 4 days. And we found that knockdown of PNPLA3 reduced the free fatty acid-induced lipid loading in just those I-148M donors, but not the wild type donors. We wanted to follow up with a more therapeutically relevant kind of treatment schedule. So we used a GalNAc conjugated PNPLA3 siRNA to reverse the lipid accumulation. So while doing the transfection method is great for target validation, this is more therapeutically relevant. So as we know, in the clinic, we're not administering siRNA as a preventative treatment. And so we wanted to have the ability to evaluate the target or the compound's effect on an established fatty liver disease. So to get around, as I mentioned, the transfection method doesn't really work in mature HEPATOPAC. We used a GalNAc conjugated siRNA. And so for this, we seeded hepatocytes, began the lipid at day 7, and then allowed the lipid to accumulate for 4 days, really establishing that disease phenotype. Then on day 11, we added the GalNAc PNPLA3 siRNA, and for this entire treatment, we continued the lipid treatment. So it wasn't GalNAc siRNA treatment and then also switched to lean media. This entire time, we had the forcing reagent. We did 2 redoses and then did the lipid staining and lysate collection at the end of the study on day 16. So for these studies, we used escalating concentrations of that GalNAc PNPLA3 siRNA, and we're able to reverse the established lipid accumulation. So looking at the free fatty acid in the middle or the combination of free fatty acid and high glucose fructose, we did see a concentration-dependent reduction in the lipid accumulation, as well as a somewhat concentration-dependent increase in PNPLA3 gene knockdown. What's interesting about this is we did not see any difference between the 2 genotype donors. Currently, we don't know exactly why that is, but it could be a result of the extended treatment period, so this was for a much longer period than the 4 days, or different dynamics when we're talking about establishing disease and then reversing it. And then I just want to close out with one more example where we looked at CIDEB. This is known to have multiple SNPs that are associated with reduced fatty liver disease risk, and these are loss of function mutations. So although we don't have the genotype for these donors because there are so many different SNPs, we used siRNA to kind of assess the effect of these different mutations. And so these mutations are known to reduce both a lipid accumulation as well as lipid droplet size. So again, we use the high content imager to measure lipid droplet size. So we're a little bit higher resolution here compared to the other images. Looking at the Bodip staining again on the right, we did have to apply basically a filter to better define those lipid droplets and define those as a region of interest for size calculation. So each and every droplet was counted as a unique object, and then we were able to quantify the radius and area of those objects. So we did find that the knockdown reduced the lipid accumulation and droplet size. So we're treating with either free fatty acid or high glucose fructose. On the top, we have the scramble siRNA, and the bottom, the CIDEB siRNA. We got pretty good knockdown for this, from 90% out to about 78-80% at the finish of this study. And we found a reduction in mean droplet area, which is on the left, and the lipid accumulation for the different media types. And we were able to put this into a distribution of size of these different lipid droplets. So we're looking at relative frequency here. There was an actual reduction in the number of lipid droplets, so normalizing it to the number, and found that the CIDEB siRNA was able to eliminate a lot of those larger lipid droplets. They did not form with this siRNA. So just to summarize what I've talked about, the HEPATOPAC co-culture system is not just for admetox purposes. It can be used for disease modeling and combining drug toxicity studies that focused on a variety of aspects of fatty liver disease. And we can specifically use siRNA to examine the influence of knockdown on lipid loading parameters. So we talked about either PNPLA3 knockdown, having different effects in genotyped hepatocytes, as well as using GalNAc siRNA in this model to look at more clinically relevant treatment schedule. And so we can use this model to test targets or examine siRNA drug candidate behavior in vitro, including different treatment schedules or assess their effects in different genotypes.